Good afternoon from Helsinki, and a warm welcome to today's science seminar, ECHA science seminar. And I'm very happy to have with us today Professor Dr. Martin Scheringer, going to tell us why persistence matters. This seminar is part of a series of online seminars with scientific content, both human health and environment, science that's relevant to ECHA's regulatory activities. What we want to do here is to discuss science, hear about scientific developments, and discuss its regulatory relevance, and see how we can take up those results within our mandate. So why do we talk about persistence? Persistence is an inherent property of a substance, and it's a key driver towards the substance fate and behavior in the environment. You could say persistence describes how well a substance is being degraded by biotic or abiotic processes. And it represents a key component in hazard and risk assessment in combination with, for example, bioaccumulation and toxicity. We call that PBT. When we combine it with mobility and toxicity, we call it PMT. We have very persistent and very bioaccumulative as a category, and we look at substances that are very persistent and very mobile. So you could say persistency, and in particular in combination with the above other properties, represents a regulatory cornerstone for industrial chemicals under REACH, for biocidal active substance and the respective products under the biocidal product regulation, for surface waters and groundwaters covered by the Water Framework Directive, and for example, for classification, labeling, and packaging, and more. So understanding persistence matters, and how much that matters, and how important it is, we are going to hear today. And for that, we have invited very senior scientists in the field, Professor Martin Scheringer. He's a very busy man. He works as a professor for environmental chemistry at the Research Center for Environmental Toxicology and Chemistry at the Masaryk University in Brno, that's in Czech Republic. And he leads a group and works as a senior scientist at the Department of Environmental System Sciences at the ETH Zurich in Switzerland. In his 30 years of research, he has worked on data methods for hazard and risk assessment with a particular focus on persistence. In addition, he's very active at the Science Policy Interface. He chairs the International Panel on Chemical Pollution, the IPCP, and he's also a member of the POP Review Committee of the Stockholm Convention. He received more than 20,000 citations so far for his more than 300 papers and a few books in addition. And he has received numerous awards for his very impactful work. And I'm very much looking forward to today's seminar and would like to hand over the floor to Professor Martin Scheringer. Floor is yours.
Thank you for the invitation to speak here, and welcome everybody to this talk about persistence, which has been a topic that is important to me, has been for many years. And I would like to share some thoughts. I hope they're also a bit thought-provoking. The talk will have three main parts. First, I would give an historical overview, and then talk about some practical aspects of determining persistence and how this can be done, and also what the problems and challenges are. And then the last part will be really about the meaning of persistence, why it is so important. And we can have some questions if there's interest after the second part of as a little interruption. So to start with, we may go back to the 1960s even, and here is a paper that many of you may have seen. It was in 1969 in Nature when Søren Jensen and others reported the finding of not just DDT, but also PCBs, polychlorinated biphenyls, in marine animals in the Baltic. And this was a big surprise and a bad surprise, because they didn't expect to find PCBs in addition to DDT. DDT was kind of expected because it was released to the environment directly as a pesticide, but PCBs were not really used for environmental application, and therefore it was a bad surprise to find them. The surprise was first mostly about the mobility about the presence of the PCBs, but then we will see now also how this relates to persistence. Here is a curve of the global production of PCBs, and we see the peak around 1970. This is an estimate by Knut Breivik and co-workers and colleagues. And here, this is the year 1969 when the Jensen et al. Paper was published. So we see that now after that publication, soon the production of PCBs went down and was ended in the early 1990s. So far, so good. But then, of course, there is now a legacy, or there was a legacy, and there still is a legacy. And that's what we see here now in this report about orca whales and other sea mammals exposed to PCBs many years after even the end of the PCB production. And this is now here where they show the whales swimming, but here now we see also what that means in reality. This was a whale, or is a whale, that was found on the coast of the UK. It had very high levels of PCB in its lipid, in its blubber, almost 1 gram per kilogram. And it was 22 years old, which means it was born after a stop even of the PCB production, but still accumulated over the lifetime of 22 years this large amount of PCBs in its body. And that is exactly the long-term burden that is caused by the persistence of a chemical. So that was the first way of looking at this, and to see what it means that we have this legacy from early use and early production, and then long after even the end of production, we have that impact. Then in the early 1970s, a totally different story started, and that's the famous paper by Molina and Roland, where they describe from their modeling and theoretical calculations that the chlorofluorocarbons may destroy ozone in the stratosphere. And then years later, this is then the second paper here by Farman and al. They found actually that this happened, and that the ozone layer was damaged by the presence of the CFCs up there. And that also led then to the Nobel Prize in Chemistry in 1995 to Crutzen, Molina and Roland. So this shows this is a totally different aspect or different impact of a chemical. It's not a toxic impact. Also, the stratosphere can be considered as a remote region. It's remote in terms of it takes long for a chemical to get there. It is high up there in the atmosphere. And then still, there can again be a bad surprise by a chemical that causes impacts and damage in that kind of environmental system. Then in the early 90s, there was more and more awareness of all of this, that chemicals are circulating globally. And there was then this very nice overview and review paper by Karl-Heinz Ballschmieder, published in the Angewandte Chemie in the English edition. And he summarizes nicely all the different processes that happen when a chemical enters the environment and then circulates widely. And also, of course, he mentions that the persistence of a chemical is a prerequisite for its global distribution. And then he made that quote that says, during the last 50 years, the prediction that many man-made chemicals would reach every corner of the global environment has become a reality. That was in the early 1990s. A bit earlier, in the late 70s, this warning was published, and it's, I think, a remarkable statement, and I will just read it. Persistent materials, because of this property, will accumulate in the environment for as long as they are released. Since the environment is not effective at cleansing itself of these materials, they will remain for indefinite periods which were not recognized at the time of their original release. The problem could become entirely out of control, and it would be extremely difficult, if not impossible, to do anything about it. That was Marvin Stephenson in the early times of chemical hazard and risk assessment. Then, a little later, in the future, in Germany, also published a similar warning in 1994. He said, in the following, reasons are given to support the thesis that persistence is the central and most important criterion to be applied in assessing the environmental hazard of a substance. In exactly the same sense as human toxicity is the central and most important criterion in assessing the hazard posed by a chemical in the workplace. So again, this idea that persistence really matters a lot because it implies a distribution, exposure, and also damage. And again, this is now more than 30 years ago, and in the future, today we are still thinking about this very hard and also struggling with the problems that have become manifest. And that's what we will turn to in the end of the lecture today. Then in the 1990s, there was more work on persistence. It was the time when I also published my first paper in environmental science and technology, where I also tried to see how we can calculate persistence on a model basis. Who was also very active was Donald Mackay and Frank Vanya, who also published something about how to calculate the multimedia persistence of chemicals that integrates all the different single media half-lives into an overall metric of overall persistence. So it was a high time of discussions about persistence and long-range transport, and it also created the foundation for the Stockholm Convention on Persistent Organic Pollutants, which was negotiated, the text of which was negotiated in the late 1990s. In the future. Then, as a last kind of stage to be released or to be started here is the PFAS part and the PFAS story, which is of course in a way the most important part of the persistence story because PFAS are the most persistent organic chemicals that we have. This started with this paper by Casey and Challen in 2001, where they reported the presence of this selection of 4 PFASs that were found in wildlife around the world: PFOS, PFHXS, PFOSA, and PFOA. As I said, I think it's appropriate to say that the PFASs are paradigmatic persistent chemicals. They are organic chemicals, which is exactly what we are talking about here, because persistence, of course, matters only for organic chemicals. Metals are persistent anyway by very definition, by their very the way they are. We are talking about chemicals that could be degraded into water and CO2 and salt or whatever. And PFASs are the ones that don't do this because the carbon-fluorine bond is so strong that it cannot be broken in the environment because there's no process that has enough energy in the environment to break that bond. So even if there are some reactions in the non-fluorinated part of a PFAS, not these ones because they are also already perfectly, completely oxidized. There's carboxylic acid and sulfonic acids, but there are others where this non-fluorinated part may be oxidized in the environment and change. But what remains unchanged is the fluorinated chain here in red. And we can say, I think we have to say, that the fluorinated chains remain in the environment forever, at least parts of them. In the future, in the future, in the future, in the future, in the future. As I said, the Stockholm Convention entered into force. It was agreed on in 2001 as a result of the negotiations in the 1990s, and then it entered into force in 2004. And it has the topic and concern of persistence in its name. It's the Stockholm Convention on Persistent Organic Pollutants. But it has 4 criteria that would define a POP, not just persistence, namely P, B, and T for persistence, bioaccumulation, and toxicity, and as the 4th one, the potential for global, long-range transport for global presence of a chemical as the result of environmental transport. These have been some landmarks in the development of this story of persistence since the 1960s. The Stockholm Convention is, which is very good, an active and dynamic in the future system arrangement. It's a multilateral environmental agreement, and it has the clause that says that more POPs can be added to the convention in the future evaluation by the POPs Review Committee. And that the POPs Review Committee has been very active and also productive in the years since the convention entered into force. And in 2009, the first group of new POPs, of additional POPs, was included. And then since then, several more rounds have occurred or have been done. And now it's the number of additional POPs is now even 25. So that's good. And currently there is an important question under the convention, and that is the question about short-chain PFAS, which of course these chemicals are still PFAS and they are very persistent. So under a convention on persistent organic pollutants, they may also matter. Here in this slide, we see some of them. These are perfluoroalkyl carboxylic acids with up to 6 fluorinated carbons and their precursors that may oxidize into or break down into these carboxylic acids. So this is perfluorobutanoic acid here with 4 carbons. 3 of them are fluorinated, and perfluorohexanoic acid here, and also materials that are precursors because they may degrade in a non-fluorinated part and then form these acids. And then it's also perfluoroalkyl sulfonic acids with up to 5 fluorinated carbons and their precursors. Again, you see here the perfluorobutane sulfonic acid and also a sulfonamidoethanol that is a precursor for that acid. And these chemicals are of course also of concern because they are obviously very stable, very persistent, and increasingly also found in the environment. And there is this question to the convention, it's an open question, are short-chain PFAS POPs in the sense of the convention? They have different properties, but they have this key property of extreme persistence. And that may imply that somehow the convention may or should even try to address and cover also these chemicals, because I think they are persistent organic pollutants. But they have different set of properties compared to what we had before. If I go back here to PB and T and long-range transport, we now have a different combination, but I would argue that the property profile of short-chain PFAS is complementary to this profile that we have here for the typical, let's say, DDT and PCB-like POPs, and that it would make sense that the convention can also cover these persistent chemicals in the future. So that is the end of my historical introduction. That shows how the topic has developed over the years. And now I will move on to some practical aspects of quantifying persistence and identifying persistent chemicals in the future. And this is an area where many of you are also experts, I think, and may even know a lot more about individual test systems and processes than I do. I just have an overview here because, first of all, that's also what I teach in my lectures to the students about chemical risk assessment. What are the most important processes that do degrade chemicals in the environment that should be captured by any test system for persistence? This is, of course, reaction with OH radicals in air, a very important and even aggressive agent, the OH radical. To some extent, for some chemicals, also direct photolysis in air, meaning reaction with light. Then, of course, hydrolysis in water, and very important biodegradation in aerobic and anaerobic systems. And in general, we need to, in the end, to estimate persistence, we need to derive half-lives of these degradation processes. And for that, we have either, or not either or, but we have as complementary systems, experimental systems, and also estimation tools that derive an estimate of a chemical's half-life from the chemical structure, based on earlier measurements from which this tool kind of learns what kind of chemical elements or groups of chemical groups that make up a molecule, how they would contribute to a chemical's persistence in the future. There are, of course, the OECD test guidelines for ready biodegradation and inherent biodegradation, and then also the guidelines for simulation tests. And many of these guidelines are also, or have been further developed, have been modified and amended. And just at the recent CTEC Europe annual meeting in Vienna last month, there was also a meeting of CTEC's persistence interest group, which I found very interesting. And I really think that a lot of very important work is done in that group, because they discuss the reliability and the viability of the test systems and also what the meaning of the test outcomes are, and also how the tests could be improved. So there's a whole world, I guess, that could also be a subject of such a seminar here, the experimental testing for degradation in the future. On the other hand, there are estimation tools, and there are quite some available of different groups who created them. But what we still often use is the set of tools used present in EpiSuite, in that set of estimation tools developed by the US EPA in the 1990s as a very comprehensive set of tools. They are available free of charge, and in a way they have been investigated a lot and also tested, and they're quite reliable within reason. And for degradation, we have mostly BioWin and AOPWin. BioWin is for biodegradation, and AOPWin is for reaction with OH radicals. It's the atmospheric oxidation program, AOP. And there are, of course, and that's also nice, that for all the components of EpiSuite, there are scientific papers, often in environmental science and technology, where the derivation of these methods is described and background is provided. And here's the paper where BioWin was introduced in 1994. Still a valid paper that I often recommend to my students, because I also recommend BioWin to them. In the future, for degradation in air, as I said, it's AOPWin, and here is a paper of the many papers by Roger Atkinson, who conducted numerous measurements of degradation in air by OH radicals and collected these data, and then also derived from these data the tool AOPWin that incorporates the results from all the measurements. In the future. Recently, we also did something related in our own team here in Zurich. That's a paper that came out just last year by a student, Paulina Körner, and my colleagues Juliane Glüge and Stefan Glüge and myself. And here we used machine learning to extract the information that is available in, let's say, all reliable, all available data on ready biodegradation. This is a classifier, so what we did is not a tool that would predict a half-life, but they would predict or estimate the yes or no as a test for ready biodegradation. So again, no half-life, but just the first step in persistence estimations, namely, would a chemical pass or not pass the test for ready biodegradation. And in that paper, we first, of course, then took a large data set for ready biodegradability of more than 6,000 chemicals. And what we found from that data set from an earlier publication was that some or many of the data were what we would call noisy. So they would have different results, yes or no, or yes and no, for the same chemical, which often happens in biodegradation assessments and is also one of the big pains in that area, because the data are often, or the conditions vary, and the bacteria react to these different conditions, and then the test result for the same chemical may change a lot, just depending on these conditions. So what Paulina Körner then did in the end was to really curate the data and try to remove the noisy data and make a smaller but more consistent data set. And then she trained a classifier on that data set, and that classifier obtained a balanced accuracy, which is a metric of sensitivity and specificity, a value of 94.2% for the balanced accuracy, which is very high, and we can kind of proudly say, for now at least, is the best classifier that is currently available, measured by this balanced accuracy. And the classifier is available online as a tool. You can see the link down here, and you can also then try it and use it yourself in the future. in the future. But this was hard work because of this, the enormous amount of noise in the data. And that is really a pain that I guess that many of us know from our persistence assessment work. That's also what we covered by this team in this paper here, by Andreas Schäffer and Katrin Fenner, Janwin Wang and myself, where we try to address this question of the noisy data and the inconsistent findings for chemicals that have perhaps intermediate persistence or low or intermediate to low persistence is not totally clear. It depends on the setting. And what we say in this paper is first, the point here up, in the first row is in green low persistence and in red high persistence are of course the easier cases. Here the assessments are often relatively straightforward and results are reliable. For low persistence, the test for biodegradability, OECD 301, is quite reliable. So if a chemical passes that test, then it is kind of very certain that the chemical is not persistent. And if, on the other hand, the chemical doesn't show any degradation in any of the tests systems that are currently existing, it's quite obvious that this chemical really is persistent. But what about all the ones in the middle? And that's a large group where the data are noisy and you may have conflicting findings in the future. I would like now to read to you 2 quotes from the paper. I know that this is not the best way of presenting in a presentation, but I think it's important and maybe of relevance to your work at ECHA. That's why I still think we may take the time and look at this quote here and also on the next one, on the next slide that's coming. So what we say in the paper is, given the complexity of the environmental factors influencing chemical degradation and the diversity of commercial chemicals, it has to be accepted so that for chemicals in the middle group, the intermediate persistence group, even improved testing methods will not remove all of the immanent heterogeneity in their persistence data. For cases with widely different but technically valid persistence data, a weight of evidence approach is necessary, and the benefit of the doubt should follow the precautionary principle in order to protect human and ecosystem health. What we mean by this is that it could be that for the same chemical, there are test results that indicate high persistence and others that indicate low persistence, but as long as they are technically valid, of course, the one that indicates high persistence should be accepted as valid and also followed in the assessment, because if it's not a measurement error, it shows that under some conditions that may occur in the environment, a chemical is actually persistent, whereas it may be less persistent under other conditions. But if there is some set of conditions that make the chemical persistent, then that should count. That's what we want to say here. And then there's a second quote, in the future. In persistence assessment, scientific logic aims for a mechanistic description of the processes involved, so real good understanding. It aims for low uncertainty and a comprehensive understanding derived from a broad empirical basis. That is the ideal of a scientific derivation. Now, if this scientific logic is used as a benchmark in the regulatory context, this may easily lead to paralysis by analysis. While regulatory decisions should be based on sound science, of course, discrepancies between scientific goals and regulatory needs and consequently different levels of requirements, must have versus nice to have, for degradation studies need to be recognized and appreciated in the future. So here we want to say that it is a different kind of logic that applies or is used in scientific studies and in a regulatory setting, which you are in, most of you, I guess. And as scientists, we always would like to know more and would like to do more and have what it says up here. So a mechanistic description, low uncertainty, comprehensive empirical basis, that is the ideal of a scientific investigation. But even if that is not accomplished, there may be some part of valid findings in the set of data, and these valid findings might be enough for a regulatory decision. So my point here really is that in the regulatory context, the bar should be put at a level that is different from where it is put in a scientific investigation in the future. And that's also something we can discuss even at this point, because this is now the point where I would ask if there are any questions at this point that we could discuss before I move on to the third part.
Yes, I was wondering from the first paper about the machine learning classifier, I was wondering whether it could be applied back to the noisy data to remove the noise, to denoise, as it were. But then immediately you presented the second paper with the 2 quotes, and basically you answered my question in that way, in a policy way, rather than scientific way. So thanks for that.
Well, thank you for that comment, and I think that is exactly what I wanted to convey. So great that you saw the point, but I can still say or try to respond to your kind of science-oriented or science-directed question. We did not do this because we were not sure that if that would lead to a kind of a circularity, but we used other sources. We used BioWin to denoise the data, but we did not go back to the same data with the new classifier in the future, informatic data. Yeah.
Thanks.
Or at least not in the improvement of the classifier. I think we may have actually done that and checked what was what the noisy chemicals would give, but of course we didn't include that in the training of that classifier.
Yeah, of course. Thanks.
Yeah.
And thank you, Kaishu, for making this an interactive seminar. Any more questions in the room or online? So everyone is eagerly waiting for the next part. So Martin.
Okay then. And I go back to my slides. I would like to hear perhaps at the end really something about what you think about my point about scientific aims and goals and scientific perfection or level and the regulatory setting and the difference between these two. That's something I would like to just learn something about from you. But for now, I go back to my slides.
And I just got reminded by a colleague that 2 more hands were raised, that I just need to find those hands. So please go ahead. Whoever has a hand raised, please speak. Panos, please, Panos.
Thanks for the slides and also for the paper. We recently read it carefully, and we were thinking with Heidi and colleagues to contact you perhaps next week to discuss some details. But for now, I wanted to ask, given the variability of the experimental test, I mean, even in the guideline, if I'm not mistaken, of 301, the extreme values of degradation can differ up to 20%, and this is still a valid test. So taking into account just the variability of the test, let alone the limitations of the training set, limitations of descriptors, bias and variance in the models, and so on, how many chemicals would be close to the 60% threshold or 70% depending on the test? And because of their variability, it's a matter of luck whether they go left or right of the threshold. So I wonder, the 94% of balanced accuracy, is it really realistic to expect that in the real world when you also have chemicals that you have not seen before, maybe they're a bit further in the training set, or even the same chemicals, just because of the variability of the test?
Yes. No, I think the 94.2% are just for really a metric that indicates the performance of this qualifier on this set, nothing more. I wouldn't say that this is something we should now expect for further, what it can expect if we change the data set. And if you include more data that are again noisy or even outside the domain, the chemical domain that was used as the basis for the training. And as you said, exactly this is the point. There may be valid results that differ by, let's say, up to 20% or even more. And then that's what I try to say in these 2 quotes, that if they are both outcomes are valid, then we should go with the indication of persistence of the higher half-life or the no in the edit test. But of course, do also further investigations and try to expand the basis for a final decision. So also run other tests and confirm if the indication that it may be persistent in the future. I guess the only way here is really to have a broader basis. That's also what we said, weight of evidence in that 1 quote. We need as much evidence as possible in a way, as reasonably possible. Of course, not too much that would lead into paralysis by analysis, but it has to be a broader picture for these chemicals in the middle group.
Yeah, agree. Yeah, thank you.
Yeah. The final part is really also what's kind of close to my heart or part of my thinking all the time. In addition to all the questions of how we do it and how we determine persistence, what does it mean and why is it a concern? And how can we express that it is a concern? And that's, I will try to do in several different ways, several different attempts, how we can first understand and then convey the meaning of persistence. So first, start from this statement, and that is something I have heard several times or many times in my life, mostly from industry representatives. That persistence just indicates the presence of a chemical and has not really any immediate meaning for actual hazards in terms of toxic effects. And I think that is a misleading statement. I think what persistence means really, and that's what we could see with the Orca whale, persistence indicates an impactful causal relationship that extends far into the future. That's what it really indicates. So even if we are here now in the present, we know that a persistent chemical will be there in the future, and we know it will cause effects also in the future. And that is, of course, an immediate indication of concern, whatever these effects may be. So even if we don't know at this point now, today, what the effects may be in the future, we should still take into account it as a given that effects will occur in the future from the chemical that's present today. In the future. A different way of expressing this is if we look at how we normally describe risk in chemicals assessment, which is the ratio of a concentration of a chemical in the environment, a certain medium often, let's say in water, and an effect threshold where a certain species or the set of species where the effects start to become manifest, or below which there are no effects to be expected, given the current knowledge. But now if we start emitting a persistent chemical and keep emitting it, then we may move from a situation that is under control, let's say, because the risk is low, because concentration is low, to a situation where concentration gets higher and higher, and in the end, we reach a case or situation where concentration exceeds the effect threshold, the same effect threshold, same conditions, same chemicals, same environmental system, same species tested, but now because of the ongoing accumulation, we have a risk. And that's, of course, obvious in a way, but I think that is very important to take into account, and we have such a case right now, which is causes a lot of discussion in Switzerland, also beyond, and that's trifluoroacetic acid. Here we see exactly this in these two charts. On the left, it's from leaves and needles of trees, and on the right, it's data or levels in groundwater. You see that over decades, and you see that steep increase. We know where it comes from. It comes from fluorinated gases used as heat carriers, and it comes from mostly also pesticides that contain a trifluoromethyl group. And so far, trifluoroacetic acid has been considered as not very toxic. At some point, of course, it is toxic, but that is a high concentration. But if we could keep going like this, then we may exactly have that situation here, that we go from the green situation to the red situation, because at some point, of course, it will be toxic. What will exert its toxicity. Let's say that it has a toxicity that is kind of inherent, and if we go high enough with the levels, then of course this toxicity will become manifest, and that's exactly the risk or the possibility into which we are running here. So this is really an important warning, and this situation exactly illustrates this concept or this conceptual thing here from green to red. And in addition, I guess you are all very much aware of this because ECHA just has published the German proposal that trifluoroacetic acid should be classified as toxic to reproduction. And then, of course, this changes the picture again, because then the effect threshold also goes down, and we have a new situation, even worse, even more alarming. But even if it's not, at some point, all organic chemicals will somehow cause unwanted effects. The dose makes the poison still. I mean, at some point we reach a point where effects will occur in some kind of organism somewhere in the world, or even in the stratosphere, as we had to learn from the CFCs. So that is this situation here, and we're exactly kind of witnessing this kind of development in the future. A third way of looking at it is this one here that I would like to expand on now and explain. The title of this slide says persistence increases toxic effects. How would that be? To see what this means, we need to consider the two factors that determine the duration and magnitude of exposure to a chemical. And these two factors are very different. One is emissions. They determine, of course, whether or not the chemical is in the environment, and they are not chemical specific. They are driven by technical needs, economic needs, and decisions by people to use a chemical in a certain way. And of course, if we are close to the emission source, then we are exposed. And the second factor is persistence. And this is chemical specific, and it is important because if the persistence is high, then this leads to a longer period of exposure and also to higher levels of exposure in the future. So in the worst case, it even makes it irreversible if the chemicals don't degrade, such as PFAS. And so here we have a factor that is separate from the emissions, and what we can then say is, for non-persistent chemicals, it's really the emissions. If we are close to emissions, we are exposed. If not, then we are not exposed because the chemical is degraded before it reaches remote regions. But for persistent chemicals, in addition to, of course, still proximity to emission sources, the persistence determines the type of exposure that people and ecosystems experience, because the chemicals may go around the world and expose people who are very far away from the emission source. And also, the exposure will be lasting and people in the future will be exposed. And that has nothing to do with emission sources, because that could happen or would happen even when the emissions have stopped. So what we can say now is, if we say the dose makes the poison, or the dose leads to the manifestation of toxic effects that the chemical has the potential for, if we say that, and we say as a second point, the persistence determines the dose. If it's very high, of course, high persistence determines the dose, then we can say high persistence makes the poison, because it leads to relevant exposure in large areas, and in the future that causes the effects that are connected with that chemical in the future. So I think we really have to see persistence as an enhancer or amplifier of toxicity. And that's also what I said here in this editorial in Science recently. In the blue box, it says persistence acts as a multiplier of toxicity. And this insidious aspect has been underestimated in many assessments, and also, I think, in regulatory assessment schemes in the future. By that I mean, in the future, higher weight should be given to high persistence in the future. The assessment scheme also, let's say, under REACH in the future. So again saying, if the persistence is high, it is much more important as a criterion, as an aspect of hazard than all the other aspects. And we don't have to go very far, I think, in the testing for effects if we can see that the persistence is so high in the future. So again, I think in a way this is telling the same story again and again and again. Here is a paper that shows what we want to avoid, namely the global spread of persistent substances. This paper came out 3 years ago. It caused a lot of attention. We were kind of overwhelmed by the waves it made. And it has, and you may have seen it, contains in one of its figures is this chart, or this panel that shows levels of P4. We compiled levels of P4 and PFOS and others from other studies. We did not do any, we didn't make any own measurements. We compiled levels from the literature, you see that here, from different areas in the world, in rain or snow, so in precipitation, concentrations in nanograms per liter water. And you see this is a logarithmic scale here, and the different colors indicate different types of regions, closer or more remote, closer to sources or more remote. And blue is really directly with next to a fluoropolymer plant. yellow or orange is urban regions, green is rural areas, and then this brown is really remote. That is Antarctica and Tibet. And then you see here this dashed line that indicates the current US EPA health requirement for P4. And we see that everywhere in the world, even in these very remote regions, the rain exceeds, or the levels of P4 in the rain exceed this line. And that is was then read, and I guess that's not written wrong. It rains toxic PFAS, or the rain is toxic from PFAS in the future. Sorry, it was just a helicopter passing by. Yeah, and that's exactly what we want to avoid, that we everywhere in the world have that situation in the future. And that was a follow-up work. You see, it's the same group of authors as we had them here. The team with Ian Cousins, Jana Johansson, Matthew Salter, and Bo Shah. We then published this follow-up piece where we we made this graph. This shows, and that's what it says in the title of this slide, persistence makes serious environmental problems. These are timelines of 5 groups of persistent chemicals, and materials, very, very different ones. You see, PFAS, plastics, chlorofluorocarbons, PCBs, and DDT. And the timeline is from the 1950s to today, and it shows with the red exclamation mark the first warning. And you'll see that in some cases, that when it was published, the first warning was published. In some cases, it was really early. Also in the plastics battle that we are fighting now, even in 1972, it was warned that plastic would kind of get out of control as a global type of contamination. CFCs, Molina and Roland, Jensen et al. We saw that earlier today, and of course also Silent Spring. So early warnings, really, and still in many of these timelines, we're still dealing with these chemicals, with these materials, and they are all global and they are all problematic because they are so persistent in the future. That's why we also then advocated what we call the P-sufficient approach, again saying that if high persistence is found in a chemical, then this should lead to regulation. Whatever exactly the toxic effect or other impacts, such as ozone depletion, whatever the other effects are, if high persistence is found, this is not just a warning, this stands for the manifestation of effects. At some point in the world, at some point in time. With this, I come to my conclusions. So as we have seen now, high persistence is a serious concern. And then the second bullet point is also very important and makes it even a new point, because if we now combine high persistence in the environment with long elimination half-lives from the body and chronic toxicity, then we have the worst case possible. And that's what we see with PFAS. PFAS have elimination half-lives, toxic or kinetic elimination half-lives from the body of, let's say, 4 years. That's very long. So even a single dose of these chemicals leads to long-term exposure because of the slow elimination. And that exactly is then the basis for the development of chronic diseases caused by PFAS. So this combination of environmental persistence, slow elimination from the body, and chronic toxicity is really a disaster. And that's what we can see now with the long-chain PFAS. And that's also a concern that was highlighted to me personally by the PFAS case, that chronic effects often show up later in people, and they are not captured by initial regulatory risk assessments, because they unfold over decades and we don't even see them in the early tests. So this is also a very insidious aspect of long-lived chemicals. So in a way, you have to go back to square one, the Stephenson quote that was very direct and very straightforward and very clear from 1977 that I read out earlier. And it says, what is not degraded will stay, perhaps even forever, in the global environment. That's what it says. And we also have seen now from the presence of the chemicals everywhere, even the volume of the global environment is not large enough to dilute away, you could call them to dilute away persistent chemicals. They don't go away, even by the massive dilution in the global system. And that's also what the Balschmieter quote was in Karl-Heinz Balschmieter's paper at the beginning today. But here this graph from the Frank Vanya, Donald Mackay paper shows this nicely, that we have the circulation of the chemicals, but they don't go away. And dilution is not the solution to pollution. I will say it can't be even for shorter-lived chemicals, because what the solution then is degradation. Degradation can be the solution to pollution, but dilution can't be. And then a more even philosophical conclusion at the end. Of course, the problem is easy to understand. This is something that is really, kind of obvious and has been known for long, since the 1970s. And that's similar to CO2. We have known for even much longer, even more than decades, even centuries, that CO2 will cause global warming. That was John Tyndall in 1861 who pointed out how massively shocked he was by the strong warming effect of small concentrations of CO2. Nevertheless, I guess we have to witness this. We see that our societies are not able or willing to act accordingly. They don't act on persistent chemicals. They don't act on CO2 emissions. But, and that's my last point, it's also an important point that I learned recently, that this is, of course, highly dramatic and concerning, but it is a political problem. It's not a problem that we as scientists or regulators can solve. It's a political problem that has to be solved politically, and if there's not the political will for it, it will not be solved whatever we as scientists or you or we together, you as regulators, do. And with that, I'm at the end, and thank you very much for your attention.
Great summary and explanation why you believe P matters, and I believe many of us will agree, or at least you provoke a lot of thoughts here in the room and online. And we have at least one comment already by Bastian. You can probably read that. So yeah, so please, everyone, don't. I can also read it out for everyone.
Should I leave my slide up or not?
I can see your slide, the conclusion 3. Yes.
Yeah. Should I leave that up or should I stop sharing?
Please, maybe leave it on for the time being. Maybe someone would like to refer to a slide.
Yeah. Okay.
So our colleague Bastian refers to the P-sufficient approach being currently tested on the PFAS, and you can probably read it. Would you like to?
I can also say it quickly as well, if you want. No, so of course you're very much aware of the ongoing work on PFAS in the committees. We already went through the opinion making and now even a decision on PFAS and firefighting foams. So I think that was an excellent first test of the P-sufficient approach. But it was just a relatively constrained sector, let's say. There was already solutions out there, similar like with the CFCs back in the day. So it was kind of easy, and now we're getting too hard with a lot of users and a lot more substances are also really being affected. And it's just interesting to see how this lack of recognition in the regulatory framework of P is now becoming sort of the argument for not regulating based on P. So it's sort of like the rat biting its own tail. So I just wanted to highlight that because we keep on hearing that that was sort of the biggest argument against the hazard assessment of the universal PFAS restriction proposal
in the future. Yeah. Thank you for this. I hear you, and I find it concerning. And I would say, just in agreement with you, there is a lack of recognition for the importance of the P criterion or the P dimension in the current regulatory system. That is, I guess, now we have to accept and have to see. And of course, we should somehow try to modify that, change that. But I think really the system has been kind of blind on that eye in the future.
Thank you, Bastian. And now Romanas would like to comment or ask something.
Thank you, Martin, for your inspiring and very interesting talk. I was wondering a bit, again, discussing on substances which are persistent and regulating on the basis of persistent. Of course, the stability of the substance which is by design, maybe depends on technical function or expectations what it would serve for. So there are some probably functions where there is an interest of having stable substance, at least for some time. Then looking into regulatory area currently or let's say legislation which is currently available, we have some thresholds for persistency to call a substance that it's persistent or very persistent. With that perspective, would you think that if substances are regulated only on a basis of persistent without considering toxicity, bioaccumulation, mobility, which is a case currently, would you think that there is also a need of thinking what would be those thresholds, whether those thresholds which are currently available, like surface water, I think it's 40 days, half-life 40 days, and if it's above, then it's persistent substance. Would those need to be reconsidered to see that it's not overly regulated versus also that it's missing some of very nasty substances which might be driven a concern just by persistence? Thank you.
Yes, thank you. So I have 2 points in response. First, on what you said about the need also for stable substances, I think that is exactly why it is a restriction, right? The restriction that also offers derogations and transition periods for exactly uses where, let's say in this case, of course, PFAS are still needed. And of course, there has to be this kind of balancing between a very important use, we may call this an essential use. I guess you have also discussed the essential use concept, at least informally. And then it's not just to say these chemicals have to be banned tomorrow. That is not also what I would advocate. And then second, the threshold, I must admit, I don't have a clear answer to this. I see your point that where is it that persistence should take over in the decisive criterion, at what level or what extent of persistence. And if it's on the order of half a year or more, or a year, I guess we may reaching that area. I'm not sure if we need something like an extreme persistence threshold of, let's say, 5 years or so, that chemicals above 5 years would be regulated on persistence alone. But of course, this has to be explored in more detail, and it's also not just a scientific question, it's also a political question. I think POPs, for which the persistence threshold is on the order of half a year or a year, are already close to something that is that should be regulated on the basis of persistence, because a half-life means that only after 5 or 6 half-lives the concentrations go down to virtually zero, to very few percent of the initial amount. So it takes much longer than just 1 half-life to really go down. Yeah.
Are there any more comments or questions? And I see Christoph has one. Please, Christoph.
Yes, thanks, Martin, for this very inspiring talk. So, I mean, taking what you are saying and putting this into a regulatory framework, would you then advocate for some sort of prioritization system where we would perhaps sort substances in a first sorting according to their persistence, and then in a second sorting according to their toxicity, maybe? So, and then basically, that would also somehow reflect some ideas that we have had in the past about, making differences between different PBTs, for instance, instead of treating them all like plain vanilla being the same thing.
Yes.
This could really kind of be interesting in terms of prioritization.
Yes, I totally agree. That would be the way I would advocate and think that it would make sense. Absolutely.
Great.
Because again, if the chemical is not persistent, then of course everything else comes into play as we know it. It's just to say that at a certain point we have to have a different regime in the future. Good, thank you. In the future.
Thank you, Christoph. So we have plenty of time left. And anyone in the room would like to add something? Online, I have one question, so if there's no one else, then I ask. Where do you see currently the weaknesses and strengths of how we assess persistence? And how do you see the future of persistence assessment, both in the lab and in silico?
Yeah, first, I mean, I'm not an expert on the laboratory tests. So I was, as I said, I attended the Persistence Interest Group meeting at ECHA. Again, I found that very interesting. In a way, this group, I think, could also be called the Degradation Interest Group, because it's really about degradation tests and how well they work and how they can be improved to be meaningful and really reliable and valid. So I think this is a very important avenue, the work that's done in that group and related work, of course, also beyond that group. But this group, it really is important to address all the technical and also conceptual questions of the existing tests. That is really very, very, in the future, important. Then what we as scientists often say is there is, unfortunately, there is not much funding on the scientific side for degradation tests. So to improve also the estimation tools, we need more data, more empirical data, of course. And it would be great if we had, if we could just run a range of different degradation tests for a wide range of chemicals in the laboratory, not as a regulatory activity, but as a scientific activity, and we would spend even, let's say, 1 or 2 years on these tests and just run a well-selected set of, let's say, 700 of chemicals through different types of tests and learn and have a better empirical basis and try to constrain that noise if possible, or to find out the noise will always be there and the noise is confirmed. But more measured data would be so good and so important. But this is something that is not sexy and doesn't attract or generate a lot of research funding in the future. But that's something I would really, really like to see. And if I had some money, I would perhaps even spend it in this way or find a partner who can run all the tests in the laboratory and organize this kind of systematic, comprehensive degradation testing. Also as a basis for better estimation tools. Because people complain, oh, you're using EpiSuite and BioWin from 1994, oh, that's outdated and old and not valid, but then give me more data and better data, then we can try develop better tools, but without more data, we can't. In the future.
Thank you. Thank you for your answer. Very, very, very helpful. And yes, also kind of sales pitch, not bad.
It wouldn't be to my benefit really, because I would not really be the lead person for that. I would just try to kind of catalyze this kind of work.
Thank you very much. Anyone else who would like to participate, contribute? Don't be shy. This is recorded, but we can also crop the recording, so don't be afraid of asking something that you might regret. So I ask a question that I don't know if I regret it. How would you like ECHA to possibly change if you could decide the way we assess or conclude on persistence?
Oh no, I mean, first of all, I think ECHA is really important and doing a lot of important work and right now there's so much criticism of REACH, and we see, I guess we all know what may happen with the REACH revision and all that. So I think ECHA should really defend the system and hold up its core elements. That is something that we all as scientists hope that this is what ECHA can do. And what I heard from what we just said before, if there's a willingness and openness to see that kind of prioritization scheme in terms of persistence first and then go back to what we have and other criteria, if that is something that is kind of agreeable within ECHA, and ECHA could even argue along these lines, I think that would be fantastic. So I see a lot of openness here from what I heard in response now to the talk. And that is encouraging. So, and that I wouldn't say, I wouldn't be in a position to express criticism really or whatever. I think what you're doing is very, very important. Keep it up, be strong, resist to all the current attempts to dilute the system, to dismantle elements of the system. That is what we have to, I guess, face and try to avoid in the future.
I don't see any hands up in the room, nor in the audience. So then I thank you very much for this excellent seminar. looking forward to the audience feedback on organization and content, and also your feedback, Martin, how we do that. With that, I would like to close the seminar, and yes, tune in again for the next science seminar. We will be back. Thank you for your time and your contributions. Thank you. Bye.
Yeah, thank you again from my side again for the invitation. I think it has been very interesting and productive, and I hope we can somehow continue the conversation. Thank you very much.